All right, I think we have kind of a critical mass, so I'm going to go ahead and start us off. Um, my name is Brigid Wills, and I am the Aquatic Reserves Stewardship Coordinator at Resources. I'm serving my AmeriCorps term here, and I'm excited to welcome you all to the annual Cherry Point Science Forum. Before we get started, oh, sorry, making sure that I can, there we go. Just a couple of Zoom reminders. This presentation is being recorded and we will be posting it on the Resources YouTube page. Just kind of general Zoom etiquette, please make sure you're muted while our presenters are talking.
Really quick.
If you watch on speaker view, that'll kind of give you the best best viewing experience. Just a little note about the way that things are going to run. We'll have a Q&A after each presentation. During while the presentations are going, feel free to enter questions into the chat, and during those Q&A sessions, we'll pull questions from the chat to ask to the presenters. You can also use the raise hand function during those Q&A sessions, but hold off on raising your hand or unmuting yourself until we're in those Q&A sessions. All right. Okay, next slide. There we go. I would like to give a quick land acknowledgement. We live and work on the ancestral homelands of the Coast Salish peoples, including Lemhi Nation and Nooksack Indian tribe, who have lived in the region from time immemorial. We acknowledge the tribe's treaty rights as well as our responsibility to uphold them, and we're grateful for the tribe's enduring care and protection of the lands and waterways. Cherry Point Aquatic Reserve, which we'll be talking about today, is located on the usual and accustomed fishing grounds of the Lemhi Nation, as well as being recognized as tribal cultural property of Lemhi Nation by the U.S. Army Corps of Engineers. Lemhi Nation was really integral in preventing the Gateway Pacific Terminal in 2016, and recently, over the winter break, BP made a purchase of Cherry Point that Lemhi Nation opposes. So I'd encourage you to read more about the tribes in our area and their involvement in environmentalism and helping us protect our shared waterways. Sorry, just making sure this is going. All right. So a quick note about Cherry Point Aquatic Reserve. It is 3,050 acres of state-owned land here in Whatcom County, and it was designated in 2010 with a boundary change in 2017. It's located near a couple of refineries, as well as some historical heavy industry areas like the Intalco aluminum smelter. And it's a really important ecological area because we have species like eelgrass, surf scoters, orcas, Pacific herring, among a lot of other ones. And it's a really important spawning habitat for forage fish and habitat for marine birds. Resources, which is one of the groups organizing this forum, is a nonprofit organization based in Bellingham. We mobilize people to build just and thriving communities and to protect the land, water, and climate on which we all depend. At this point, I'm going to turn it over to Robert to give a quick introduction of the Cherry Point Aquatic Reserves Citizen Stewardship Committee and talk a little bit about what they do. So Robert, go ahead and take it away.
Thanks, Brigid. My name is Robert Kaye. My pronouns are he and him. I'm a member of the Cherry Point Citizen Stewardship Committee, which is really just, and so first of all, thanks for showing up. Thanks for coming, and welcome to yet another wonderful science forum. This is the 2024 version, and we've been doing this for quite a few years now. And of course, thanks to all the presenters as well. A little bit about the committee. It's a loose ad hoc committee of people who either live in the area or even if you don't live in the area, they have a great interest in the Cherry Point Reserve and all of the Salish Sea in and around. It's, as Brigid said, it's quite a special place. I'm sure you all visited there and know it quite well. Some of you know it probably a lot better than I, since you're actually doing work there. But the Citizens Committee is really tasked with a few different things. It's under the auspices of the DNR and the Cherry Point Reserve. And we act together to do things like marine bird surveys. We do intertidal surveys. We've done a lot of community research over the years. We have different events. We put this event on. We also do a wonderful event in the summertime, and usually almost always in June, at a low tide in June, called What's the Point, at the Cherry Point Reserve. And if you're not familiar with that, and you like what you see here today, I highly encourage you to come out in June to What's the Point, because it's a very special event. We have naturalists on the beach, and people get right down, and you can look at all the critters under a little scope, and it's just a wonderful day. If you are interested at all in what we do, I'd encourage you to go to the website. We're always looking for more people to participate. We have a really good group. It's fun. Actually, it is fun. I've been doing this a long time now. And the people who are involved are great people. And I think it's worth your while to spend one meeting a month for a couple of hours. It's a great thing. So I went on a little more long-winded, as I always tend to do, but thanks again for coming, and look forward to this wonderful presentation from our scientists. Thanks.
Wonderful. Thank you so much, Robert. And yeah, the CSC is just a really wonderful group of people, and I've had the pleasure of working with them for a couple months now, and we'll continue doing so through the summer, and I'm really excited for that. So thank you again to our speakers. I'm just going to give a quick intro of who we're going to be hearing from and roughly what time we'll be starting. Again, we're going to have a Q&A after each of these, so you can have your questions then. And we are going to be hearing first from Olivia Fletcher, who is with the Department of Natural Resources Puget Sound Corps, talking a little bit about eelgrass. Then we're kind of going to have a chunk of European green crab monitoring information. So we'll hear from Lindsey Parker from Washington State Department of Fish and Wildlife, as well as Leah Skare and Allie Simpson from Northwest Straits Commission, and they will be all talking about European green crab monitoring. We'll just have a collective Q&A session at the end of that. And lastly, we'll hear from Allison Brownlee from Department of Natural Resources about Cherry Point herring and how DNR has been assessing larval and juvenile presence. So at this point, I'm going to turn it over to Lyle Anderson, who is another member of our Citizen Stewardship Committee, to introduce Olivia, our first speaker.
Good morning. I am Lyle Anderson, in fact, and so I'm honored to introduce Olivia Fletcher. Olivia went to college in Florida, where she is originally from, where she majored in biology and was introduced to her love for seagrass through an undergraduate research project. She moved to Washington after she graduated to pursue a career in marine science, which is how she landed in the Puget Sound Corps. She is currently leading the program's seagrass monitoring projects. And please welcome Olivia.
Hello, everyone. I'm going to attempt to share my screen now. One second. Okay. Okay, can everyone see the first slide of my presentation?
Yeah, that looks great, Olivia.
Okay, great. Okay, so as you know, my name is Olivia, and today I'm going to be taking everyone through the details of our seagrass monitoring projects and discuss where we are in our data analysis process. So for those of you who don't already know, the Aquatic Reserves program was established in 2004 by the Department of Natural Resources, and just 6 years later, the Cherry Point Aquatic Reserve was established. And our ultimate goal is to promote biodiversity and to protect critical aquatic habitats. And now you may be wondering who the Puget Sound Corps are. Well, we're a team of AmeriCorps members that work for the Aquatic Reserves program as essentially field technicians, and we run on 11-month terms. They like to call us their boots on the ground, but during the winter months, we spend more time in the office because on top of field work, we also do a lot of work on outreach projects and data and lab analysis. So our team focuses on 6 main long-term monitoring projects across the 8 aquatic reserves. And you can see the projects for each reserve outlined in this table. And each Puget Sound Corps member is in charge of at least one project. I, for example, oversee the seagrass monitoring, which takes place across 5 aquatic reserves. We do regularly participate in projects that are outside of these 6 main ones that you see here, like the Cherry Point Light Trout project, for example, which you will be seeing a presentation on later today. So the Aquatic Reserves program's biggest focus is on our long-term monitoring projects. And generally, when talking about ecological monitoring, long-term is considered 10 or more years. And this is so a baseline can be developed to understand the initial environmental status of an ecosystem. And these projects are essential for understanding ecological responses to environmental change. And the real idea behind long-term monitoring is that it can provide a lot of insight, not only to the health of the ecosystems, but to the effectiveness of current management efforts and future management actions. So seagrass can be found on every continent of the world except for Antarctica, and it's a very valuable ecosystem for many reasons. One of them being that it can actually remove carbon from the atmosphere at a rate that's only rivaled by salt marsh and mangrove ecosystems. And 1 hectare, which is about 2.5 football fields, is estimated to be worth about $19,000 US. And on top of that, seagrass also supports commercial fisheries by providing habitat for a wide array of species, as well as protecting coastal communities by stabilizing the shore. So there are 72 species globally of seagrass species, but the 3 species that our monitoring focuses on is native eelgrass, non-native dwarf eelgrass, and toothed surfgrass. And the 2 main species that we find at Cherry Point are native and non-native eelgrass. So consistent methods are critical for global comparison and analysis, especially when dealing with a species like seagrass, which is found all over the world. So we chose a data collection method that is a part of the worldwide seagrass monitoring effort led by the Smithsonian Institute. And these methods include collecting both environmental and biological data at our sites, which allows us to get more of a full story about what's happening with our seagrass beds. And so this slide paints a picture of our field collection methods. And what we do for each reserve where we monitor seagrass is we have a designated 50-meter long location that lies on the zero tide line that we refer to as a transect. And we lay 50-meter measuring tape out along the transect and then use 1/4 meter squared quadrats to take density and seagrass height measurements at designated meter marks. And then we take photos of the seagrass in the quadrats in case we want to do a visual analysis. And speaking of, so this is an example of what a visual analysis would look like. Right now, the only story that we have is based on what we simply see is happening visually. And as you can see from these photos, we have quadrats that have never had any seagrass in them, like quadrat 12 on the left. And then we have quadrats that have shown an obvious visual decline in density, like quadrat 3 on the right. But our transect is not actually indicative of how the entire Birch Bay seagrass population is doing. So as you can see from this picture, our transect covers a small and specific part of Birch Bay seagrass. And right now, we have no way of knowing if seagrass has been lost from the bed as a whole. And this is because we only monitor intertidal seagrass, which completely comes out of the water for parts of the day. So we really can't speak on how the subtidal seagrass is doing. And we can only say that observationally, we have seen reductions in seagrass along our transect. And we know that many of you are probably eager for an analysis beyond visual observations, and trust me, we are too. We're currently working on doing quality checks and cleanups on the data that we have so we can feel confident in the numbers that we're attaching to our visual observations. And this can be a somewhat tedious process. I mean, we have been to this site 44 times in the last decade or so, and dozens of Puget Sound Corps members have handled the data. So a lot can happen with the data that requires extensive checks and sometimes corrections. But we're very hopeful and excited to get it all cleaned up and analyzed. But as for a timeline, it is hard to say, but we are working on it constantly and have regular meetings about it. So getting this information to the public is definitely a priority of ours that is really only preceded by the priority of making sure we're giving you accurate information. Okay, so this chart right here does a great job illustrating the interconnected nature of specifically eelgrass and the surrounding world, and why the question of what's happening to the seagrass is such a complicated one. And this chart shows possible eelgrass stressors on the left and how those stressors connect to the factors controlling seagrass bed health in the middle, and then the biological aspects of seagrass that are used to measure health. And the boxes indicate the data that we collect, with green representing environmental and blue representing biological. We collect numerical values for everything except herbivory, which is really just used more as supplemental information to our data analysis. And so, as I mentioned, we do our seagrass monitoring at 5 different aquatic reserves, and the stressors that you see here are largely site-specific, which is why we can't take a one-size-fits-all approach to our data analysis. And that, of course, would save us a lot of time and effort, but it would, in the end, compromise the integrity of the findings, and that's the last thing we want to do. So we're trading time for accuracy in this case. And that's my presentation. And thank you everyone for listening and giving me your time. Does anyone have any questions for me? Oh, I see a hand from Lyle. Okay.
Yes. Have you seen any evidence of the, I guess it's called seagrass wasting disease?
Not that I'm aware of. We haven't seen any on our transect specifically. But if you want, I can give you someone's number to reach out, but as far as I know, we haven't seen any evidence of that at Cherry Point, at least.
Okay, thank you.
Okay, someone just asked in the chat, any general observed changes over time in native versus non-native eelgrass in the area? And I believe, yes, we have seen, so the non-native eelgrass is called Zosera japonica, and I believe we have seen it kind of creeping up into our transect, while the native eelgrass, which is Zostera marina, is kind of creeping down more out of our transect and more into parts of the beach that are more covered by water for most of the day.
Olivia, I have a question. Could you talk a little bit about the site-specific stressors that you see up at Cherry Point?
Yeah, so that's kind of what our data analysis is trying to show us, is like, what are the site-specific stressors? I can imagine that there are some anthropogenic stressors, like people just going out and walking on the beach. Maybe there's a lot of boaters, but for the specific stressors, it's hard to say without our full data analysis. Sorry. Yeah. We'll get that information out there as soon as we can. Okay.
Thank you.
Yes. So, okay, someone asked in the chat, has the state done any planting of seagrass similar to the plugs of sea oats in the dunes in coastal Florida? If I correctly recall, I believe on the aquatic reserves, we did try a restoration project at one point. And I know that actually just recently, the legislator announced millions of dollars that will be going into kelp and seagrass restoration. So those efforts are definitely going to be kicking up in the next few years. The question is, the Cherry Point seagrass meadows healthy? Well, I can't officially say that because, as like I showed in my presentation, our transect measures such a small and specific part of the Cherry Point, because the intertidal seagrass can be so different from the subtidal seagrass, which is, I don't know if I can pull that back up, which is what that drone footage was showing. I'm going to try to pull that picture back up. Hang on 1 second. Well, I'm trying to. Okay. So as you can see, like this red line shows where we measure. And then you have all this green on the left, and that's more of the subtidal seagrass. And just from this picture, it's like, oh my gosh, look how much of it. That's so great. But we just don't know for sure because we don't have the capacity to monitor that seagrass specifically. But we can say for certain how the seagrass, at least the intertidal seagrass, is doing once we've officially completed our data analysis process, and that information will be made public once we're done. Are any local industries such as BP interested in your findings? I would like to think that they're interested, but I don't know. I don't have any interaction with BP, and I'm not sure if the Aquatic Reserves program has much interactions with BP. I'm sure if we find something that's like alarming, we would let the public know, and then BP would probably find out through that. And did you notice an impact from the heat dome of 2021? I wish I could answer that question. I started my term in 2023, so I can't say, but I do know that it wasn't such a clear, like, oh my God, the seagrass is all gone. And the seagrass does fluctuate throughout the year. Like in the summer when it's hotter, the seagrass it goes down, the density of it. And then when it's cooler, and the tides are at night, so they're not exposed to so much. The low tides are at night, so they're not exposed to the sunlight when they're out of the water, that theoretically the density would be better. But again, once the data is out there, we'll know for sure. Temperature is one of the things that at least I'm working on analyzing right now. And we're just as interested as you are to get that information. So we'll get that out to the public as soon as we're done. And temperature is a priority of something we're analyzing.
Sweet. We probably have time for one more question if anyone has one. Oh, sweet. Yeah.
Are there optimal temps for seagrass to survive and thrive? Yes and no. It really is species dependent because, as I mentioned, seagrass is on every continent except for Antarctica. So it can do really well in like very cold Alaska, like conditions in Alaska. I know it thrives in Florida. For Zostera marina specifically, I think that it does like the average temperatures of Washington State, but I'm sorry, as I keep mentioning, we just can't say for sure what's happening, at least with our seagrass. I believe there is a kind of a range for Zostera marina, Zostera japonica. But I'm not sure how the changing in temperatures over the last couple of years, with temperatures rising in Washington, if that's impacted the eelgrass specifically at all.
All right. I don't think we have any other questions. Thank you so much, Olivia. That was great. Thanks so much. It's great to hear what DNR has been working from. At this point, I'm going to turn it back over to Robert, who will introduce our European green crab speakers.
Okay, I'm back. How's everybody doing? Well, that was really fascinating. I see that stuff out there all the time, and it's always amazing that they're doing those studies. It's really great stuff. I will just quickly read what I've been given on these bios and let you get into a huge issue that everybody has a lot of interest in, and that's our green crab issues in the Salish Sea. And this is starting, first presenter is Lindsey Parker with Washington Fish and Wildlife. Lindsey set her first trap for European green crab back in 2016, when efforts were focused on preventing and detecting the first inland green crabs. With a background in invasive species management and marine science education, she worked both with Padilla Bay, NERR, I don't know that one, and the Washington Sea Grant Crab Team to continue early detection green crab efforts in the Salish Sea before joining Washington Fish and Wildlife's Aquatic Invasive Species Unit in 2020. As the EGC, or green crab population, has increased, so has Fish and Wildlife's efforts to protect our native species, and Lindsey's passion for this project has also increased exponentially, right? Lindsey is currently the regional European green crab biologist responsible for the coordination, collaboration, and trapping. I hate it when my phone does that. In Whatcom, Skagit, and Snohomish counties, and her large team of technicians is active in all those areas. Then we're going to hear from members from the Northwest Straits Commission, Leah Skare and Allie Simpson. Leah serves as the Ecosystem Project Specialist for the Northwest Straits Commission. She supports the work of the Northwest Straits Initiative on a variety of regional projects, including the monitoring and removal efforts of invasive European green crabs in Drayton Harbor, and she's been doing that since 2021. Allie Simpson is an ecosystem project coordinator with the Northwest Straits Commission. And at the Commission, she works on a variety of projects, including European green crab management, transboundary eelgrass protection, and efforts to improve diversity, equity, and inclusion in marine sciences. Yay. Before joining NWSC, the Straits Commission, Allie completed her master's in marine and estuarian science from Western Washington University. So that's a mouthful, and all I can say is, all three of you, thank you so much for doing these presentations, and we're thrilled to have you, and we're honored to have you, and take it away.
Sweet, thanks, Robert. Lindsey, if you want to go ahead and share your screen.
Sorry about that. I apologize. I'm trying to get it to share real quick. Of course, it works so smoothly before the meeting starts, so then there we go. Is that working for you all?
Yeah, we can see your whole presenter view.
Got it. There we go.
Okay, there we go. Thanks, Lindsey.
Perfect. Thanks so much. So, Hi, everybody. My name is Lindsey Parker. Like was said before, I am the Regional European Green Crab Biologist for Washington Department of Fish and Wildlife, working within the Aquatic Invasive Species Unit. So today we're just going to look at specifically Whatcom County and a little bit of how that fits into the bigger picture statewide. So for starters, I wanted to give a very brief overview of what a European green crab looks like and what it doesn't look like. Green crab have a very distinct characteristic that our native species do not share, which is 5 marginal teeth located on each side of the carapace, just behind the eyes. They also have 3 rostral bumps between their eyes and somewhat flattened rear legs. And their carapace is kind of oval or pentagon shaped, really going from that wide to narrow. We get asked a lot about how big European green crab are, and they're larger than our native shore crab species, but smaller than our typical recreational crabs like Dungeness and red rock. So about 4 inches is our typical max size on them. And one thing to note is that European green crab are not always green. Their undersides can range from yellow to orange to red, and the tops of their shells can be mottled brown, black, and green. Also to note is that a lot of our native crabs are green and commonly get mistaken for European green crab. So on the right, I have some examples of some native crabs that are commonly mistaken for European green crab. So at the top here, we have hairy helmet, kelp crab or spider crab, hairy shore crab, and purple shore crab, which also are not always purple. So again, these are not European green crab. And none of them have those 5 marginal teeth. So that's really our key way to tell them apart from our native species. In Washington State, WDFW, or Washington Department of Fish and Wildlife, is the lead state agency for the management of European green crab on the waters of our state. Tribes have jurisdiction on their sovereign lands and federal agencies on federal lands. European green crab are classified by the state as prohibited level 1 species, which is the classification that poses the highest invasive risk and that are the priority for prevention and expedited rapid response management actions. So European green crab are considered one of the world's worst invasive species. As an estuarine species, they have small larval stages. They've been easily transported to new coastlines through human-mediated pathways such as ballast water in seafood and bait packaging materials. They're extremely prolific and voracious predators that aren't picky. They're very tolerant of range temperature and salinity conditions, all factors which really make them successful at establishing in areas outside of their native range. This illustration, courtesy of Washington Sea Grant, shows on the left a healthy nearshore ecosystem. And on the right shows the potential dramatic ecological impacts that dense populations of green crabs could have. In areas with extensive invasions, they have been blamed for devouring shellfish resources, decimating eelgrass meadows, and altering the structure of our shoreline. We are particularly concerned about the potential risk they have in the Salish Sea because of our native oysters and commercial oyster populations and their competition with Dungeness crabs and their impact to eelgrass nursery habitats that could have cascading impacts to salmon, shorebirds, and the people who depend on these resources. So really big issue. We're definitely really concerned with the impacts they could be having. Looking at Washington State in particular, European green crab in Washington are not new. They were first detected in 1998 on the coast of Willapa and Grays Harbor. Initial management actions took place following the discovery, but due to lack of evidence of self-recruitment and funding resources, the project fizzled out over a few years. But then in 2012, a new population of green crab were discovered in Sook Basin, Canada, which is just across the Salish Sea from Dungeness Spit. So that detection warranted more efforts once again in Washington. So 2015, WDFW designated Washington Sea Grant to lead an early detection monthly monitoring community science network, also known to many of us as CRAB Team. Following the launch of the CRAB Team network in 2016, we had our first discoveries within Washington's inland waters at Westcott Bay in the San Juan Islands and in Padilla Bay. The following season, in 2017, there were new detections at Macaw Bay in the Macaw Reservation and Dungeness Spit, located in the Dungeness National Wildlife Refuge, which is managed by Washington or U.S. Fish and Wildlife Service. This graph looks at the number of green crab removed over time. And as you can see, there were really large increases in about, going from 2016 to 2017, we started to see large increases. And then again in 2021, the shift from about, just under 7,000 to over 100,000 European green crab looks really scary on a map or on a graph. But one thing I'd like to caution all of us is to think about effort as well. Anytime we look at just captures and not any sort of effort that was tied to that capture can really skew our results. So anytime that we have increased effort, we're going to find more European green crab, which hopefully helps continue to find funding and resources so that we can continue to increase our effort. And so that's one of the things that we'll talk about a lot in likely mine and Leah and Allie's presentation is catch per unit effort, which is a way that we quantify it using a rate of crabs per 100 traps is the kind of standard that we use to compare so that we're factoring in how much effort it took to find those crabs instead of simply just number of crabs being found. Another great thing about Washington State is we have so many partners that are interested in putting an effort towards this European green crab issue. Here in Whatcom County, we have the Northwest Straits Commission, Lummi Nation, the MRC, our Conservation Corps members, Department of Natural Resources, and many others that are all really interested in keeping our ecological impacts that could happen from European green crab at bay. One thing to note is that I will be presenting on Department of Fish and Wildlife's data, and Leah and Allie will be talking about Northwest Straits, but there are other agencies like Lummi Nation who have their own data source and data set that I won't be speaking on since that's not my story to tell. Here's a map of our different management areas. There's 2 management branches, the coastal and the Salish Sea, which is broken up into 14 management areas. And particularly today, we'll be talking mostly about that North Puget Sound area. So statewide, since 2022, over 640,000 crabs have been removed from our shorelines. Key areas that those have been removed from were Grays Harbor, the North Puget Sound, and Willapa Bay had the largest portions of that. I will say that this is a screenshot from a live updating dashboard that I'll send the resources out at the end. I'll put them in the chat. But our 0 for 2024 is because this is combined monthly, and so our month isn't up yet, so I'm sure that won't be 0 for much longer. There's a lot of people hard at work still removing green crabs. Out of those 646,000, 361,000 crabs have been removed in 2023. And then zooming in to the North Puget Sound, out of our larger picture, 86,000 crabs have been removed from the North Puget Sound region. In 2020, that was just over 80,000. And then this year, even with increased efforts, 2023, we captured just over 6,000 crabs. So seeing trends in the correct direction. So zooming into Whatcom County in particular, there's three main sites, three main water bodies that I'll talk briefly about. And then once again, just wanted to note that Lummi Natural Resources and Lummi Nation have their own data about what has been happening on their tidelands and in the Lummi Sea Pond that I won't be touching on yet. So one of the sites that has been trapped historically is Birch Bay, which is obviously the closest to the Cherry Point Reserve. And so 2019, no effort was conducted, but 2020, European green crab were found in about 50 trap efforts. So going back to our catch per unit effort, which is crabs per 100 traps, we're looking at a roughly 2 catch per unit effort. Because that one crab was captured, the next year we came back and did a much larger effort. So a lot more traps were put in the water and still only one more green crab was captured. So our catch per unit effort went quite a bit down. So overall, over those 2 years, we weren't surprised that we were catching green crab, and our concerns were fairly low, just because the population seemed to be relatively low. No effort was conducted in 2022, but in 2023, we did go back in and trap. Department of Natural Resources and Northwest Straits Commission helped do the first effort in June and caught the first European green crab this year. So wanted to shout out that Aquatic Reserve staff were a part of that effort. And because that green crab was captured during that time, 5 efforts over the season were done in Birch Bay in particular. Most of those crabs were captured out of the mouth of Terrell Creek, seen here. So all those red dots are where green crab were detected. One change this year that resulted in our kind of increased catch rate was also a shifting our trap type and really targeting shrimp trap placement compared to some of our other trap types in the mouth of Terrell Creek. So all but one of the crabs we captured this year were in a shrimp trap. So 97% of our crabs were in shrimp traps compared to one crab that was found in a minnow and none that were found in our fukui traps. Leah will touch a little bit more on the different trap types in her presentation, so I didn't want to take time to do that here, but just briefly, our different trap types are used in kind of to target different types of populations and in different scenarios. So shifting our trap type here in Birch Bay really did help remove green crab. And then shifting a little bit south to Bellingham Bay, there's kind of 2 spots that we've captured European green crab here in Bellingham Bay. So first, Post Point by the Post Point treatment plant has been a Washington Sea Grant crab team site since 2016 and has been consistently trapped by Department of Fish and Wildlife since 2019. And until this year, had not had any European green crab detections. This was the first year, so it is a new detection for 2023. And 4 European green crab were captured from there. And because we've got the first European green crab in June. 3 additional follow-up resulted in those 4 crabs. So our catch per unit effort is still relatively low, since we really did try to hit that area hard with traps to really maintain that we felt good that the population was low. Bellingham Bay is kind of a catch-all for a lot of these sites, but the thing to note is that while a lot of Bellingham Bay has been trapped, the only place that has captured European green crab is right in this corner of the Whatcom Harbor. The other kind of interesting thing about the data for this area is that the Marine Life Center here in Whatcom Harbor has a educational trap that is unbaited, and so has captured European green crab, but it's not in the same way that we conduct our normal trapping efforts. So it's not really comparable to put that in our catch per unit effort. So our first real detection was by a Marine Life Center trap for European green crab back in 2021. And because of that, we really went in and did a large-scale effort throughout Bellingham Bay to assess what the population looked like, and did remove 3 more European green crab that year. Still, our catch per unit effort was relatively low at 1.43 per 100 traps. And so since then, anytime a European green crab has been found at the Marine Life Center, we've done a small follow-up effort to make sure and get a pulse check that populations aren't increasing too much. But there has been kind of a consistent low-level population in this area. Chuckanut Bay down to the south. I will also say that these 2 outside points at Clark Point aren't included in this data. They're actually included in the Bellingham Bay data, since they're not inside the trestle there. But the Chuckanut Bay has been a Washington Sea Grant crab team site since 2017. And the first European green crab was found in 2019. 3 European green crab were removed, still maintaining a 1.35 catch per unit effort. Trapping was conducted in 2020 and 2021 without any more European green crab removal. And only 1 green crab was found in a pretty large efforts that were done in 2022. This year, we did see an increase in our catch per unit effort up to 5.91. So a total of 23 European green crab being removed there from this year. The asterisk by our catch per unit effort is we did catch a European green crab by hand. So similar to how we're not including that in our numbers from the Marine Life Center, it's not really comparable because you can't include that as a per trap. So that's why that asterisk is there. And just to kind of look at them and be able to compare all of our sites next to each other, currently our highest CPUE this year was Birch Bay. And part of that is because of our shift in kind of looking at more removal efforts instead of early detection efforts using those shrimp traps. But overall, all of our sites did increase in European green crab detections for 2023. And looking into 2024, part of what we're looking at doing as Department of Fish and Wildlife is looking at long-term monitoring in the Salish Sea. Washington Sea Grant's crab team does a great job at early detection monitoring and has a really great long-term monitoring project for that. But what we're looking at doing is including our shrimp traps that have been found being really effective for removal efforts and less sensitive for early detection efforts, because we're really hoping to answer the question of if our removal efforts are having impacts. And so that would be the way that those 2 efforts would complement each other. We're also, our team up in Whatcom County is looking to continue our partnerships with Department of Natural Resources, Northwest Straits Commission, and Lummi Natural Resources to really help dig in and do assessments in the areas where European green crab have been found, and continue to look at early detection efforts throughout our region. To wrap up, I do have a whole array of European green crab resources. So instead of sending a whole bunch of links, I have one link that I'll drop in the chat that's for this cheat sheet that is a collection of links for anything that you might be interested in. So we're holding questions till after Leah and Allie's presentation as well, but thank you so much for letting me speak today, and I'm excited to hear more.
Thanks so much, Lindsey. That was great. And a note about that link, we'll make sure to send that out in a follow-up email as well, so people have that to reference to. And Leah and Allie, if one of you wants to share your screen, go for it. Yeah, and reiterating what Eleanor just put in the chat, feel free to put any questions in the chat in the meantime, if any popped up while Lindsey was talking, and we'll circle back to them at the end.
All right, I will go ahead and get my screen shared. Does that look good?
That looks great. Thanks, Allie.
All right. Yeah, so I'm Allie, and I'll start off our presentation today, and then I'll turn it over to Leah. And I want to just thank, I can't remember the person's name who gave us the introduction, but I really appreciate that. So I wanted to start off today by actually giving you an introduction on who we are at the Northwest Straits Initiative. The Northwest Straits Initiative is an umbrella term that encompasses 9 different entities. So 7 volunteer-led county-based marine resources committees, the Northwest Straits Commission, which is the part that Leah and I are with, and then the Northwest Straits Foundation, which is our nonprofit partner. Our role in the Northwest Straits Initiative is to provide coordination, funding, and technical support to the marine resources committees and connect their work to other important regional initiatives and emerging issues, so like the green crab work. And we work with 7 MRCs, like I mentioned. So this is the 7 MRCs within the initiative, but specifically for green crab, we work closely with Whatcom and Skagit MRCs. And the MRCs the beauty of the MRCs is that they bring together a diverse perspective from the community. So things like tribal, economic, environmental, and recreational interests to advise local government on marine resources issues, educate the community, and carry out resource protection projects. And lastly, our mission is to protect and restore marine waters, species, and habitats of the Northwest Straits to achieve ecosystem health and sustainable resource use through a local-based approach. And it's really that local-based approach that helps sets us apart and the lens through which we really conduct all of our green crab work. And that's important because, well, green crabs don't see borders. I think Lindsey did a really great job of kind of describing all of the impacts green crabs have. And what they see within the Salish Sea is just miles of shoreline habitat in which they could invade and potentially impact. And for us at the Northwest Straits, we think that increasing trapping and outreach efforts through a collaborative approach could help limit the impact green crabs could have within the Salish Sea. And so I'll talk about our trapping and outreach efforts in just a second, but I wanted to highlight the areas in which we are actually working in and how that can relate back to the Cherry Point Aquatic Reserve. So with the Northwest Straits, we are focused in on 2 hotspots within the North Sound, Drayton Harbor in Whatcom County near Blaine, Washington, and then Salish Bay, which is in Skagit County. And Cherry Point sits kind of right in between both of these. And like Lindsey mentioned, green crab can easily get to new locations. And with them being in Birch Bay and with a hot spot of green crab activity in Drayton Harbor, Cherry Point is an area of concern for green crab potentially being in the area. And with that, I wanted to go back to talking about our the targeted outreach that we've been doing in these areas. And so we've worked really closely with the Marine Resources Committees to provide targeted outreach about green crabs to the local community. And specifically, I wanted to highlight some of the efforts that Whatcom MRC has been doing that really we could not do without their help. And this goes back to that collaborative approach. We can't do all of this by ourselves, and we really need to work with members of our community and different partners to be able to get this done. Since we've been working on this project in 2020, Whatcom MRC has done amazing outreach efforts, including sending an educational mailer on green crabs to over 1,000 residents within the Blaine area. That was hugely impactful for us. Not only were we able to get the word out about what green crabs are, but we were able to let neighbors and shoreline landowners know what it is that they may be seeing out in the bay. They may be seeing a lot of people and traps out, and just being able to get the word out about what's happening was something we could not have done alone. And so that was a huge effort led by the Whatcom MRC. And then just even hosting public webinars about the status of green crab in Whatcom County. The more that we get information out there, the more people know what to do and what to expect and what's happening with this issue. And that's, again, these collaborative efforts are incredibly important and something that we're really proud of to be involved with. And so that's one arm of some of the work that we're doing is the outreach efforts. And the other part of the work that we do is the trapping efforts. And again, collaboration is key when it comes to this type of work. And I know Lindsey touched on that as well. But for this season, we worked with more than 40 different trappers in Whatcom and Skagit counties. We worked with the Whatcom and Skagit MRCs, Washington Sea Grant and Crab Team, Washington Conservation Corps, Department of Fish and Wildlife, Department of Natural Resources, oyster companies like Drayton Harbor Oyster and Taylor Shellfish, and a really great program called the Veterans Conservation Corps. I wanted to highlight a little bit more about the Veterans Conservation Corps program because this program helped us immensely out in the field in Drayton Harbor. And really, we wouldn't have been able to do a lot of the trapping effort that we were able to do in that area without this program. And so the program helps get veterans interested in environmental work and get experience in environmental fields. And so we were really lucky to be able to connect with that program and have an intern for the summer to help add capacity with our trapping efforts. And to us, that's incredibly important because not only are we including multiple members of the community that may not always have a say in this type of work, but just making sure that we are getting all of those different perspectives involved and making sure that this is truly a collaborative effort. And with that, I wanted to turn it over to Leah to actually start talking about our management strategies and the data that we actually collected out in Drayton Harbor.
Hi, everyone. Thank you so much for having me. And yes, I will dive right into the data and share a little bit about our management strategies and talk about how all of those amazing people and the 40+ trappers that came out really were integral to collecting all this data. So a big thank you to them. So tackling this problem means that it's not just one strategy, and adaptive management is really integral to our process. These management strategies include the geographically broad scale early detection programs to rapid response to localized management with repeat assessments and intensive trapping. And that is really what our work focuses on in those two hotspots that Allie mentioned in Drayton Harbor and Salish Bay. So that heavy removal, the goal of heavy removal is trapping the population down to levels that reduce European green crab abundance and their chance at those negative ecosystem impacts. So our management strategies include core site monitoring, targeted hotspot removals, and we conduct land-based trapping from April through October during low tides. So we check those traps daily, and then we remove them after 2 to 4 soak nights, typically. And so from our trapping, we can learn about European green crab relative abundance, seasonality, their invasion history in these local areas, and reproduction and growth patterns. And then we also get a really great baseline understanding of the native species composition in the area, because we're catching a lot of different species, and then we count everything and record everything and then release everything that is not a European green crab. All right, and so moving into our data, to start, this is Drayton Harbor has had a dedicated removal trapping program in partnership with all of the folks Allie mentioned earlier, especially Washington Department and Fish and Wildlife and Sea Grant. And so that's been happening since 2019 after a local Blaine resident reported a molt they found while walking the beach in Drayton Harbor. And so she reported that molt, and we were able to come out and do a rapid response and find out that this was a hotspot. So for 2023 data, this graph shows the cumulative number of captures throughout the 2023 trapping season, separated by trap type. So you can see the big orange section, we caught 84% of the European green crab in shrimp traps. And so you can really see that that targeted hotspot was really effective at the removal portion. And then the minnows and fukui traps were really effective at monitoring over time. And I'll show a little bit more about how we do monitoring throughout the seasons and across sites. But overall, we have a overall CPUE of 5.8 throughout the season. And then in addition to the 143 green crabs we caught, Drayton Harbor Oyster Company also found and removed 16 green crabs in and around their growing equipment. And so working with them has been really great in order to make sure we're removing as many green crabs as we can from Drayton Harbor. So our core site monitoring, we now have 4 years of this data set of seasonal core site monitoring. And the core sites allow us consistent locations for traps, and then we use comparable methods over the season, and that allows us to track capture rates over time and across sites in a really standardized way. And that gives us insight into what is happening with the local green crab populations throughout our time doing this intensive management. And so on the left, the photo is just a top-down example of the core site setup at our Pillars site. And so that's somewhere we go back repeatedly throughout the season in order to track populations of green crab. All right, and so on the next slide, we've got the core site monitoring. We've got each of the 3 sites I showed on the map in the last image. So California Creek, Pillars, and Dakota Creek, and then the years for each of those sites. And so California Creek and Pillars were both trapped for that monitoring in 2020. And they had a really sharp decline between 2020 and 2021. Conversely, Dakota Creek was not trapped in 2020, and it had its highest capture rate in 2021 compared to the other years it's been trapped. Throughout most of the season in 2022, we had really low capture rates at these monitoring sites, but it rapidly rose with a recruitment surge of young of the year crab cohort in the fall. And so that is why you see the increase in 2022 at California Creek and the Pillar site. And then in 2023, there was a decrease at all 3 sites. And then just over the time of monitoring these sites, you can see kind of the general downward trend, even with the surges of young of the year crabs and recruitment. And I'll go into a little bit more about how we track that recruitment and what we can understand from all the data we collect from each crab. So we measure each crab that we collect, and then that allows us to understand more about the patterns in our capture rates and assign them to age classes and cohorts based on their date of capture and their size. So starting on the left is what we call a young of the year crab, and that was caught in October of 2022, and that was during that recruitment surge. And then in June of 2023, you can see that same cohort is now a year one crab, and this crab had grown to 39.5, and it was holding eggs, so it was already sexually matured even as a year one crab. By August, that cohort, we caught a crab that was 63 millimeters. And then on the far right is an example of a year two crab, or a year two plus crab. So it's 76 millimeters with, barnacles covering it and missing limbs. And when we graph all of these crabs that we've measured throughout the time, since 2019, you can see all of the seasonal captures throughout each year, and you can see clusters forming of young of the year crabs. So that cohort is separate from the rest of the crabs, and you can start to see what percentage of our captures each year were from that young of the year cohort. And so in the first year of detecting this population, young of the year crabs made up 90% of all the captures. And then this past year, young of the year were only 3% of the captures. And so a much higher percentage of those captures were year one crabs from that big recruitment pulse that we saw in 2022. And then we also saw a bigger recruitment pulse in 2020 and 2019. And so you can really see how even in the springtime in 2023, we were catching the cohort that we had seen in the previous year. And all of that goes to, we're still learning a lot about the best management strategies in order to, understand how effective we're being and remove as many of the green crabs as possible. So we're still learning a lot, and that is where the adaptive management comes into place. So our intensive and targeted removal trapping in Drayton Harbor over the past years really does seem to be showing some population suppression, and we hope that that suppression will lead to reduced spread to other local areas, as well as resource protection within Drayton Harbor. And then that alone is a really awesome accomplishment. So in the last 5 years, we removed 918 European green crabs from Drayton Harbor that are no longer able to cause damage and reproduce. And we will continue our collaboration with other partners and continue working with local groups to make this effort happen. And that leads me to how you can help. It's always such an important piece of these presentations, is to make sure that you all go away with how you can be a part of this important project, because that is huge. So if you happen to walk the beach and you learned how to ID your green crab, please take a photo. If you find a molt or a live green crab, mark the location you are, and then submit the report to Washington Fish and Wildlife. And then please leave the crab in place. If you have access to shorelines, marine shorelines, this has been super important for us and Fish and Wildlife to be able to access new areas to kind of do the pulse check, as well as if we do find green crab, we can go in there and do ongoing management if needed. And we've had some really great volunteers come out in the field with us in both Drayton Harbor and Salish Bay. And so if you'd like to volunteer in the field, please reach out to us. We'd love to have you out there. And then lastly, there's some really great other volunteer programs, Crab Team with Washington Sea Grant, and Washington State Extension with a new molt search program. And so that is a way to volunteer on your own time. And it's a really, easy access volunteer program that helps with early detection on our shorelines, and a really great way to get involved. And so this is a molt search update from Washington State Extension, and Cheryl gave this update to me. So as of 2023, over 300 molt searches have been submitted in Washington by 115 community members. And the top 3 reporting counties were San Juan County, King County, and Skagit County. And so Washington Sea Grant and WSU Extension are continuing to collaborate on this program. And if you'd like to participate in any of the public trainings or are interested in learning about the Train the Trainers opportunity, please contact Cheryl Lovato-Niles, and her email is up on the screen, and we can add it in the chat as well. But there are some great upcoming events for you to attend to be trained on these volunteer opportunities. And then additional resources, we have some great links to learn more about all of these wonderful European green crab efforts that are happening in the North Sound and the Salish Sea. So we will add all of these links to the chat as well. Thank you. I think we are done and we can start with questions.
Wonderful. Thank you so much, all of our European green crab presenters. At this point, I think what I will do is I will read out of the chat, and then between the three of you, you can decide who would like to answer it. Before I take the ones from the chat, there was one question from the Zoom registration, and they asked, for Lindsey, has DFW explored any subtidal trapping efforts for European green crab?
Yeah, we have. We have done subtidal trapping efforts in Drayton Harbor as well as Salish Bay. And both of those were not successful in finding European green crab. And so that's definitely something we're looking more into. But from our experience on the coast, subtidal trapping is really more effective when populations kind of reach critical mass and European green crabs start moving into deeper waters. And so it's a lot less sensitive for early detection and a lot more effective when we are doing that removal efforts.
Cool. Thank you for that. This might be best suited for Leah, or if anyone else wants to answer it. Why are fukui and minnow best for long-term monitoring, but shrimp traps are best for removal, is what Kim is asking in the chat.
Sure, yeah, the minnow and fukui traps are what we started with in this program. So we have a really long data set that we can compare back to since the start of crab team and the start of trapping in Drayton Harbor. And so we didn't start using shrimp traps until 2021. So that long-term piece is a little bit of comparative to past years, as well as the opening size of the traps allow for smaller crabs to enter the trap. And so we get a really much more sensitive detection on the small size crabs and what's happening with the population of those smaller size. And yeah, if Allie or Lindsey have anything to add to that, please jump in.
Yeah, I will also say that with things like shrimp traps, we tend to use them differently than we have with minnow and fukui traps, where the shrimp traps, we purposefully put them in areas where we know there are a lot of crabs or that we've detected a crab before. And it also has more openings. And so we do use them in a different way than we do the minnow and fukuis, which I think adds to that increase of captures.
I think the last thing I'll add to that is that it's capacity-wise, shrimp traps are a lot harder for a single person to carry out in the field since we are doing land-based trapping. And so we can, one person can carry 15 minnow traps or 15 fukui traps, but might only be able to carry up to 3 shrimp traps. And so there's just, it's a good to use a mix of different traps.
Sweet, thank you all. All right, our next question from Chris. If found, why leave the green crab in place? Why not remove it?
I can take that one. Yeah, at this time, we're really not asking public to kill suspected green crabs. It may sound counterintuitive, but it's really intended to protect native crabs from cases of mistaken identity. And as a prohibited species, it's actually illegal to possess a live European green crab in Washington unless you're on our permit. So it might be really hard to let a crab go when you're concerned it could be invasive, but keep in mind that we respond to any confirmed sightings really quickly. They go straight to our inbox and we normally call you pretty rapidly about it. So the best way that you can help is report your possible European green crab sighting to us as soon as you possibly can. I don't know if Leah and Allie have anything they want to add to that, but.
Cool. Okay, at this point, if anyone wants to raise a hand and ask a question, now would be a good time. Give it a little time to let any other questions enter the chat. And while we're waiting on that, just to highlight as well, Eleanor put a molt search training in, and Cheryl is running that one, and that is specifically with Cherry Point, the CSC. And so if you're interested in getting involved in that, then that would be a great opportunity to meet some of the CSC members, and as well as learning how to do molt search. All right, Lyle in the chat, are European green crabs found exclusively in muddy habitats?
Yeah, I can take this one too. No is the short answer. So larval spread, for the most part, occurs just via ocean currents. So theoretically, a European green crab can end up along most of our shorelines. But muddy habitats appear to be, based on their history, appear to be the most suitable habitat for really creating those hot spots. So places out on like Gray's Harbor, they're finding European green crab along rocky shorelines. And we've found a few along the jetty out at Drayton Harbor. But that's not really where they can have the biggest impact, because that's not their ideal habitat. So most of our targeted efforts are for where they might have the biggest impact and where they might really get a foothold.
Leah or Allie, anything you want to add to that?
I guess I'll just say that some of the hot spots that we target within Drayton Harbor are these, it's muddy habitat around, but structures within the bays. And so there's a couple old derelict piers or an old derelict dock, and the green crab really seem to congregate there for just to hide in the structure. And so those are things we look for when we're targeting green crab habitat and trying to find the best spot to set a trap.
Super interesting. All right, our next question, Rick in the chat is asking, is there a northern cold water limit for European green crab distribution?
Allie, you can take this one if you want.
Yeah, I was trying to think of, because there have been green crabs that have been found in Alaska. And so, I mean, technically there is cold water limit for green crabs, but with climate change and oceans getting warmer, we're starting to see more of a spread further north and further into areas like Alaska. And so I think that distribution is growing, and that limit for cold water is getting a little smaller. They are incredibly tolerant species, and so they have a very wide temperature range to begin with. They can tolerate colder and warmer waters than most of our native species, and so their ability to get into new areas is just going to be a lot greater than things like Dungeness crab. And so we will be seeing them expand into other areas as well.
Got it. All right, next question in the chat. What native bird or other species prey on European green crabs?
I don't know if we have official research on this, but like other crabs, I think native birds and other larger crabs can prey on larval or small green crabs. I don't know how much that impacts their population levels though. And they're pretty feisty crabs. So even though they're small, they've got some big claws that might deter some predators. There's some interesting research on competition between Dungeness crabs and green crabs and how they might be competing for food out on the coast. I don't know too many details on that though.
There's also been anecdotal evidence that seagulls have captured some green crabs. There hasn't been, actual scientific evidence, just people saying that they've seen that. But yeah, that's something that I think would be really interesting to look more into to see. But yeah, we don't have specific evidence for it yet.
Got it. Lindsey, anything you want to add about predators or?
Yeah, I think I know there's some people doing research on seals and otters in terms of their eating European green crab. And then I know that we've done some correlation studies on how when red rock crab or Dungeness crab are in our traps, we're less likely to see European green crab. But correlation could also just be that there's, big crabs in a trap and a European green crab doesn't want to go inside there. So whether or not it's because those are actually maintaining lower populations of green crabs, or if it's just the impacts of them being in our traps is would need a more scientific study done on them.
Wonderful. I think unless there are any other questions, maybe last chance for questions, we will go ahead and move on to our last speaker. Last chance. I don't see any raised hands. All right. Well, thank you, Leah, Allie, and Lindsey. It was really great to hear about kind of the current state of European green crab, as well as just hearing about how we can help. And again, we'll send out those links at the end when we send up our follow-up email, so people have those to refer to. Sweet. Okay, at this point, I am going to introduce Allison Brownlee, who is our last speaker, who will be talking about Cherry Point herring. So Allison Brownlee is a Washington native, but has a diverse marine ecological background after working in and teaching marine biology across the globe. She did her undergraduate work at the University of Colorado. Then, after several years of teaching marine science in the Florida Keys, California, Bahamas, and Costa Rica, obtained her master's degree at Nova Southeastern University in Florida. After researching and monitoring ecosystems in New Zealand and California, Allison has landed back in Washington and has worked for the state, Department of Ecology, and Department of Natural Resources since 2017. Allison, I will hand it over to you to share your screen and tell us about Cherry Point herring.
Okay, how does that look?
That looks great. Thanks.
Perfect. Okay, thank you for having me at this forum. I am Allison Brownlee, and I work for the Washington State Department of Natural Resources as a scientist in the Aquatic Reserves program. And I'm going to tell you today about a project we've been conducting in collaboration with the Lummi Natural Resources Department and also with some guidance from WDFW, where we have deployed larval light traps around Birch Bay to assess larval and juvenile herring presence in the area. Let me get my clicker working. Perfect. So Pacific herring have a very strong ecological, cultural, and economic importance to Puget Sound. Herring are a vital component to the marine food web as they transfer energy from the lower trophic levels, such as plankton, up through to the higher trophic levels, like marine birds who forage on the herring eggs, and is also like salmon and marine mammals who prey on the juvenile and adult population of herring. Herring also have a very strong cultural importance to the coastal communities, especially to the indigenous Coast Salish peoples who have been harvesting herring for centuries. And herring also has an economic value. Prior to the early 1980s, there was a pretty significant commercial herring fishery that stopped in the early 1980s, primarily because of large population declines in herring from a wide geographic scale. There was a small spawn-on kelp fishery in the 1980s and 1990s, and this is where kelp blades were hung in spawning grounds and any eggs that attached to those kelp blades were then pulled up and harvested. But this stopped in the late 1990s. And since then, there has only been a very small sport bait fishery for herring. And this has been limited to areas south of Sandy Point in Puget Sound. So I just wanted to go a little bit about the life history stages of Pacific herring. They are a relatively short-lived species. They live to be about 10 years old. And once a year, between the months of January and May, adult spawners come into nearshore waters. These adult spawners are aged between usually 3 to 7 years, and they aggregate in these nearshore spawning grounds for a matter of weeks to months. And while they're there, they're an important prey species for those larger predators that I mentioned earlier. And then at a certain time, they all move near shore and have this big spawning event, like seen in this lower photograph on the bottom left. This is an aerial photo of the Cherry Point coastline taken by Mike McKay in 2019. And you can see that white band along the shoreline there. That is a result of the herring going near shore to spawn. And these eggs are attached to submerged vegetation, like in the photo on the left. You can see these little tiny clear to yellow eggs attached to this brown algae. They can also attach to red algae. They can attach to seagrass, kelp, any really submerged vegetation. And these eggs incubate for around 10 days. That can be dependent on temperature before they hatch and turn into swimming larvae. These larvae typically remain near shore, but they are not very strong swimmers, so they are really at the mercy of the currents. And once they get a little bit bigger, a couple months later, they turn into juveniles where they start to form schools, and they stay in the nearshore environment until about the fall when they move offshore into deeper waters. Once they go offshore, they stay out there for the next 2 to 3 years before they come back to their natal spawning grounds and join that spawning aggregation. So while they're gone for those 2 to 3 years, it's been thought that some populations of herring stay within the Puget Sound area and some populations go offshore to the outer coast. There has been studies done on chemical components of the Cherry Point herring in particular, and results from that have suggested that the Cherry Point herring do not remain in Puget Sound, and they do go outside to the outer coast. And I'm just gonna touch a little bit on their preferred diet. These herring feed on phytoplankton and crustacean larvae when they're pretty small, and then move up into more planktonic crustaceans and small fishes, but really prefer these copepods as shown in this lower center photograph. So in Puget Sound, WDFW has defined 21 different stocks of Pacific herring, and this is mostly based on when and where they spawn. 2 of these 21 stocks are genetically distinct, and this is the Squaxin Pass, which is down in the far southern reach of Puget Sound, as well as the Cherry Point stock, which is in the far northern reach of Puget Sound. Cherry Point stock is unique compared to other stocks within Puget Sound because the Cherry Point stock lay their eggs on the nearshore habitat at a much later time than all the other stocks in Puget Sound. So you can see this figure on the left. This is from WDFW, and it lists the 21 different stocks, and these green horizontal rows represent the time period for when they find eggs along the shoreline for each of these stocks. And these red boxes denote when that peak spawning is. So you can see if you look from top to bottom, most of these green bars range within the months from February to March. And then if you look all the way at the very bottom, that is the Cherry Point stock, that row that is shifted to the far right. So the Cherry Point stock, their peak spawning period is much later, in a period of April to May. And I want to point out in this figure that the row or 2 above Cherry Point, that is the Semiahmoo Bay stock and the Samish Portage Bay stock. And these 2 stocks have geographic spawning grounds that overlap the same area as Cherry Point. So they utilize the same, at least historically. So they utilize the same shorelines, they just spawn at different times of the year. But interestingly enough, those green boxes do overlap. You can see the end of March and early April. So if an egg is found on the beach on April 1st, it really is unclear whether that is from a late spawning Semiahmoo Bay fish or if that is from an early spawning Cherry Point fish. And this is when it comes in handy when this Cherry Point stock is genetically distinct. And so we can tell the difference between stocks. In this area of Cherry Point, it's really beneficial having these multiple spawning times it creates a secondary resource wave for the predators that rely on herring being present at that time. So if you can imagine these winter aggregations of the Semiahmoo and Portage Bay stocks, they come in near shore, they hang out in the spawning grounds and become important prey for larger predators. And then they deposit their eggs on the beach, which are important prey resource for different marine birds and such. And then when that's done, there's this whole secondary resource wave of the Cherry Point herring coming in, aggregating offshore. And these become important prey for species such as Chinook salmon, which are coming in and out of the Fraser River and Nooksack rivers at that time. And then they deposit their eggs on the beach and they become important prey for species such as surf scoters who are in the area at that time. So having this biological and genetic diversity supports the whole healthy ecosystem, and it has historically supported coastal communities who rely on having multiple harvests throughout the year. And it also benefits the species as a whole in the face of environmental change. And I just wanted to highlight work that was done by Eleni Petru and her team in 2021. They did a study looking at the genetic structure of Pacific herring from Puget Sound all the way up into the waters of Alaska. And so this figure on the far right is pulled from that publication. And the map on the left shows all the different areas that they sampled herring from, and they're colored by their spawning time. So the darker colors represent those earlier spawners in February, March, and then as the colors turn into yellow and orange, those represent the later spawning groups of May and June. So you can see on the map, if you look at the border between British Columbia and Washington, you see that orange circle, and that is from the Cherry Point stock. And then as you can see, they are not the only late spawning herring in the area or in this widespread area. You can see that there's another cluster of yellow and orange in the middle of British Columbia, and there's also a orange circle up in the far northern reach of the waters of Alaska. And so when they ran the genetic analysis on these samples, which is what's showing on the figure on the right, you can see that these different colors cluster out differently, which means that these late spawning stocks are really reproductively isolated, meaning that they don't really commingle with other stocks. They are isolated by the time that they spawn. So populations with different spawning times are genetically distinct. Okay, so on to the sad story. Annual populations of Pacific herring, just like other marine fish, have natural variations from year to year. But looking at a longer time scale, we can see that the overall population of Pacific herring in Puget Sound has declined over the past 50 years or so. As shown here, this is a WFW figure, and it's showing the black line on the top is the just total biomass of Pacific herring in Puget Sound. And what's important to note here is that the blue line is the Cherry Point stock, and then we have the green is Squaxin Pass, which is that other genetically distinct stock. And then the orange line is all other stocks combined. So you can see in the late 1970s, that blue line of Cherry Point stock made up over half of the total biomass of all Pacific herring in Puget Sound. And this decline in this black line follows the trajectory of the decline in this blue line, which means that the decline in the Puget Sound herring has really been because of this decline, massive decline in the Cherry Point stock. And it has declined to a point where now in 2023 was the first year that WDFW found zero evidence of Cherry Point stock spawning. So this is a map zooming into the Cherry Point area. You can see this green shaded area In the middle of the map, that is the boundary lines of the Cherry Point Aquatic Reserve. And the map spans up to Drayton Harbor in the north and down to Lummi Bay in the south. And this is a progression through time. So you can see in the 1970s on the left, and it goes to more recent years of 2021 on the right. These red circles are where WDFW has found herring spawn, and the size of the dot is relative to the estimated spawning biomass. And the yellow dots are where they sampled but didn't find any spawn. So you can see this progression from left to right of red to yellow, meaning that the amount of spawn has decreased, and then it's also contracted. So in the 1970s, the Cherry Point stock were using the entire Cherry Point shoreline to spawn on, and now in recent years, spawn has only really been found around Birch Head. So onto our project. The Cherry Point Aquatic Reserve has a management plan goal to protect and help recover species of the reserve. And we know from these WDFW spawn surveys that eggs are being laid on the shoreline. Maybe not this last year, but in the past, eggs had been laid on the shoreline. But we don't know what happens after that. We don't know if these eggs are surviving, if they're hatching, if they're contributing to the population. So we wanted to try to get an insight on the early life history stages of herring in Birch Bay. So we use these larval light traps to document presence, distribution, and growth of these early young larvae and juvenile herring around Birch Bay. And then we also wanted to run genetics on these samples to determine how many are actually from that Cherry Point stock. And then because these light traps attract all different types of critters, we had secondary objectives where if we were to catch any Dungeness crab larvae, we wanted to contribute that data to the Pacific Northwest Crab Research Group, which is a Salish Sea wide network where there's lots of partners that have light traps all over looking for Dungeness crab larvae. And we also just wanted to document what other larval fish and invertebrates that are utilizing Birch Bay during the herring spawning season. So we built these light traps to float at the surface, and they are tethered via an anchor to the bottom. These traps are deployed in about 12 feet of water, and they work by every night a light turns on, an LED light source turns on, and larvae are then attracted to that light source. So in this picture on the far left, that is what a typical light trap looks like. We have our bucket on the top, which contains the flotation and some other bits and pieces. So the water line hits just about the bottom of that white bucket, and then everything below that is underwater. So at night, the light turns on and larvae swim into those white funnels and become trapped. And then every 2 days, we go and pull up these light traps and rinse them down, take off that cod end at the bottom, and process all the contents on the boat. So we look at what species we find, we look at how many are there, we do a lot of measurements, all that good stuff. So between the months of April through June, we have deployed these light traps in 3 locations around Birch Bay for the last 3 years, so 2021 through 2023. We have put 1 trap off the southern part of Birch Head. We have put 1 trap sort of within the inner reach of Birch Bay, and then 1 off of Point Whitehorn. And I want to briefly say that side of Point Whitehorn, especially shown on this map, experiences some really strong current exchange. And we had a hard time keeping that trap there. That trap really liked to kind of travel by itself. It went up into Canadian waters once, the time it went south and went along the Cherry Point Reach. So we did end up taking that trap and moving it just around the point there and into the bay a little bit. So on to results. I'm going to first start by just going over the herring results from this project, and then I'll go into some of the results from those secondary goals of looking at what else we caught. So in total, we caught a total of 111 herring across the 3 years at all these different sites. And And before I go into this plot on the right, I want to throw out a caveat that in 2021, this was a really strong pilot year for us. These light traps we used are much different from the light traps that are used in the Dungeness crab network. In that network, these traps are tied to a dock. It's nice and calm. It's easy to process because you're on a dock. This project, they were anchored offshore in really sometimes violent conditions. And so we had a lot of adaptations we had to make through time. So this plot on the left is showing basically the successful number of hours that we were able to fish. So we have time or year on the x-axis and the successful hour of fishing on the y-axis, and then just colored by trap location. So you can see in 2021, those dots are a lot lower than the other years, and that is because we just were learning how to make these traps work really well in offshore waters. And especially that orange point in 2021 is much lower, and that's from the point whitehorn trap, which, like I said, like to travel. So we had some issues that we fixed, and by 2022, we got much better. We still had some issues with our anchoring system at the Birch Bay location, which we then figured out by 2023. And this last year, we had really high success rate. We didn't have any data gaps. So we finally solidified these methods. So the plot on the right is showing the total number of herring that we caught for each light trap location, and it's just paneled out by year. So we have 2021 on the left, 22 in the middle, and 2023 on the right, and these bars are just colored by location. So in 2021, we caught the most herring out of all 3 years, and that we were only caught them at that Birch Bay, that inner Birch Bay site location. Whereas in 2022 and 2023, we caught herring at all three sites. And you can see in the legend, there's this little red box that says submerged. And that is because in 2023, we tried out creating a small submerged trap. And the thought process behind that was, well, maybe these smaller herring are just staying down in the vegetation and not coming up to the surface to come into our trap. So we created a light trap that we could sort of sink down, and it would sit right on the vegetation, in hopes we would maybe attract more larvae that way. But as you can see, there is no red on the plot, and that's because we didn't catch any herring in that submerged trap. So looking at the size classes of these herring that we caught, this plot on the left, the x-axis is showing total length. So this is the total length of the fish. So from tip of the snout to tip of the caudal fin, total length in millimeters. And the y-axis is showing year. And then these are sort of grouped by site. So those top grouping is from Birch Bay, the middle grouping is for Birch Head, and then the bottom grouping is for Point Whitehorn. One thing to note for this plot is that because these are measurements, these are only the fish that we were actually able to measure. Throughout the last 3 years, we caught lots of fragments of herring, or herring that didn't have a complete head or a complete tail. And so we were not able to measure those fish. So like at Birch Head, it shows that we only caught herring in 2022, which is not correct. We also caught them in 2023, but just none that were measurable. And then if we look at these vertical dashed lines, those represent sort of the transitional phase from larvae to juvenile. So anything to the left of that first vertical line, dashed line, are representative of the larvae size class. Anything in the middle between those lines are in that like sort of transitional phase. And then anything to the right of that second line are going to be considered juveniles. And this is a density plot, so you can think of it kind of like a histogram. Histogram. So just along the row that the data is at, it's showing where the highest density of points lie. So what did we find most of? We find most larvae, most juveniles. So if we look at this in Birch Bay, and we're looking at 2021, you can see this in this sort of red hill. Most of the herring that we caught were in this transitional size class of larval to juvenile. Whereas in 2022, we caught a few larvae, but mostly juvenile. And in 2023, we caught almost all larvae and just a few in that transitional phase, and no juveniles. In Birch Head, we caught mostly larvae, and in Point Whitehorn, we just kind of caught some of every size. So some interesting things that I take away from this graph is that at Birch Head, which is the location where these herring are mostly spawning, is mostly where we're finding larvae. And then at Birch Bay, because of the presence of these multiple size classes across the years, it means that this inner part of Birch Bay may be an important rearing area for these early life history stages of herring. So on to the big question. Of these herring that we caught in these light traps, are any of them from the Cherry Point stock? We have run genetics on our 2022 samples. We sent in 41 herring that we collected from May 2nd to June 17th. And we had analysis help from Eleni Petru, who I mentioned earlier, who did a genetic stock assessment of herring in Puget Sound and beyond. And so she took our results and compared them to her genetically distinct reporting groups that are the January, February spawners, the March, April spawners, and then these May spawners, which represent where the Cherry Point herring would fall into. So our results came out that we had a presence of all three of these reporting groups, these spawning groups, in our samples, but 82% of them were from this March, April spawning group, which are most likely from the Samiamu Bay or Portage Bay, Salish Bay stocks. And then we had 15% from the January, February group, and only 3% from the May spawning group. So the May spawning group is what correlates to our Cherry Point stock, and 3% of our 41 samples means that we had one fish. So one of our 41 samples we sent in were actually from the Cherry Point stock. So these results were obviously a little disappointing to us, but it also brought up some really interesting questions. Because if you think back, we say, well, that's weird because we caught a lot of small fish. So I wonder when those eggs were laid along the shoreline. Was it before April 1st or was it after April 1st? And so now I'm going to walk you through some plots that we have made to help us take a best guess at that. Can we look at our samples and can we guess when those eggs were laid on the shoreline? So I'm going to walk you through a series of plots that's going to set this up because it's kind of a confusing graph. So this is, we have time along the x-axis from April through to May or June. And we have that total length from the whole length of the fish on the y-axis. And they are grouped by site. So this is 2021 data, and we only caught herring at Birch Bay. So you're only seeing this one panel for Birch Bay. And these green dots represent the individual fish that we measured and when we caught them. These gray vertical lines below it are just a visual aid to help you see when those fish were caught. So you can see that in 2021 at Birch Bay, we caught herring over 2 sample days. We caught majority of them in this early May date, and then we caught this one smaller fish later in May. So then what we did is we applied this sort of backwards growth rate. We used a mean growth rate of about 0.5 millimeter per day. And the best way that you can think of this is if you picture a juvenile herring in your head and you sort of play the video backwards. So it's getting smaller, 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 and then it turns back into an egg and it sits there and incubates, and then you can see it being deposited by the adult spawner. So these black dots on the bottom of the plot are when that adult spawner deposited that egg. So that is, those black dots are our estimated best guess at when these eggs would have been laid. And then we add in these vertical dashed lines, which are when WDFW went out and found spawn on the shoreline. And WDFW, as a general rule of thumb, uses April 1st as a guideline to denote whether these spawn they find are Cherry Point stock or not. So generally, anything before April 1st is categorized as one of the other stocks, and anything after April 1st is called a Cherry Point herring. So if we remove all of these orange lines that fall to the left of April 1st, so all of those green dots probably belong to the earlier spawning stock, and we just leave the ones to the right, we can see in 2021, there's a good chance that that one small fish that we caught in late May could have been part of the Cherry Point stock, just based on our best guess of when that egg was laid. So now if we take that plot and we apply it to the other years, this is the same plot, just 2022 data. And again, it's grouped by site. So we have Birch Bay at the top, Birch Head in the middle, and Point Whitehorn on the bottom. And we can see that there are several of these after April 1st potential egg deposition days, or days that the eggs were laid on the shoreline. And this gets really interesting because in 2022, this is the year that we sent these samples in for genetics, and only one of these fish was from the Cherry Point stock. So the question arises of, well, who are these other potentially later spawning herring? And this gets even more interesting when we look at 2023 results, where we found herring, we caught herring at Birch Bay and Point Whitehorn. And you can see that we have several of these points after April 1st, and some of them even go into mid-May. So I think this is potential evidence that hopefully Cherry Point stock actually did spawn this year, and perhaps just went undetected by the WDFW spawn surveys. So we did use a couple other techniques to look for herring throughout this project. Like I said, we put this submerged trap in at Birch Head. It was on a pulley system, and we checked it the whole season and did not find any herring. We did surface plankton tows in 2022. We did this once a week around each light trap location, just to check, are there herring in the water that we're missing with the light trap? And then we did not find any herring in the plankton tows. We actually didn't find any larval fish at all in the plankton tows. And then Erica Blakey and I also did some snorkel observations off of Birch Head, where we snorkeled around the vegetation and looked for any evidence of eggs or larvae or juvenile, and we did not see anything. That was only done on one day, but so before I go into the results, oops, I think I clicked early. Before I go into the results of our secondary goals, I just want to kind of recap on our summaries for just the herring that we caught in the light traps. So we found that our light traps were pretty successful in capturing larval and juvenile herring as a whole. And this site sort of tucked in within Birch Bay, it caught herring across all the different early life history stages, and they were caught across all 3 years. So this might serve as an important sort of accumulation area for these current driven larvae. And I'm going to show this video that was created by Terang and his team with the Salish Sea model, and it is simulated surface currents for the period of June 1st through June 16th in 2020. So this is, you can see the days at the top, so it's just ticking through each day. And you can really see how these surface currents could push larvae around in this area, and how we get these kind of big pulses that come through and really push those surface currents into that inner Birch Bay location. Our study confirms the importance of having genetic analysis and looking at the different stocks that utilize Birch Bay, especially in regards to that before April 1st. However, it gets tricky because genetics are expensive and it requires the actual removal of the fish, which when we have such a depressed stock, the removal of fish is kind of hard to take sometimes. And then we also found maybe potential evidence that Cherry Point stock did spawn in 2023. Okay, so onto our secondary goals. We found lots of different species of larval fish in these light traps. We caught over 20 different species. And along the left-hand side of this plot, you can see these are the species or species groupings that we were able to identify. And we have just total abundance, so all sites, all years combined. And you can see that top bar, that's Pacific sand lance. So we caught way more Pacific sand lance than any other species in these light traps. And our next sort of top abundant species would be gunnel, greenling, sharp-nosed sculpin, prickleback, lingcod, and flatfish. And looking at the high catches of sand lance, I started thinking, well, sand lance like to school, and I sort of wanted to remove the impact of just a school fish happened to come through the light trap. So this is a plot of looking at the frequency that we caught these species. So this is the percent of our sampling days that a species was present in the light trap. And you can see, so these are different panels. It's a lot. You just kind of have to take it out, take your eyes out from the looking at the details. But they're grouped by year and by site. And if you look at these red bars across the top, those are Pacific sand lance. So Pacific sand lance was still the most frequently caught species across all years and all traps, except for that submerged trap on the bottom, which is understandable because that was in a totally different part of the water column. That trap caught mostly gunnel, including adult gunnel and tadpole sculpin. But this plot is an interesting way to, we could try to look at species diversity for the species we were able to identify. So if you kind of look at just the number of colors you can see in each panel. So if we look between the sites, which are, if you look across the rows, we can see that Point Whitehorn has the most colors. So it has the greatest species diversity than any other site. And as we saw in Olivia's talk earlier, the Point Whitehorn trap is actually in that area where these extensive seagrass beds. And so I think that these results just show how this habitat is really providing an important nursery ground for many different species of larval fish. And then if we look across the columns, 2023, for whatever reason, had the highest level of species diversity. And that could be just due to our high fishing success rate over time. And on to Dungeness crab. This was a surprising part of our study. And I just want to start off by saying that larval Dungeness crab are in their water column from April all the way through to September. And this study really focused on the herring. So we only had these light traps deployed from April through June. We missed 2 whole months plus of catching larval Dungeness crab. So we can't use this data to apply it as a total abundance in the area, but we can look at larval delivery times, and we can look at these early pulses of Dungeness crab. That being said, so this plot on the left, we have time across the bottom, and then we have CPUE, which Lindsey earlier introduced us to. We use it as catch the Dungeness crab per hour of fishing. So per hour that light turned on at night. And if you just look at the scale change for the 2021 plot, that scale goes up to 6,000. So that's 6,000 Dungeness crab per hour that the trap was in the water. We had these massive pulses come through in 2021 at that Birch Bay site. The largest pulse was almost 53,000 larvae in just one night. And just in this short time span that these light traps were in the water, this site had a higher abundance than any other light trap location as part of the PCRG network that had fished for the entire season through September. So this is remarkable and really points out that Birch Bay is really important for lots of different types of larvae, and it's really important for larval Dungeness crab. And then you can see in the other years, 2022 and 2023, those CPU valleys are much lower, but you can also see that the delivery time was much later, which was consistent with which PCRG saw at other sites. But that doesn't mean that those large pulses didn't come once we took those light traps out of the water. It just means that we probably missed those bigger pulses. And our total abundance that we caught for across the 3 years was up near 115,000 Dungeness crab larvae. Pretty impressive. In addition to the biological parameters that we collected as part of this project, we also attached temperature loggers to our light traps. They were attached to the anchor lines at about a meter below the surface. And this plot, I really just want to highlight how much white space there is. And that is because it was really, really hard keeping our temperature loggers. And this is due because these light traps are in this offshore environment where they're battered by waves, they're battered by logs and other debris that come through. They're ripped around with the currents. And so our logger retrieval rate was pretty low, unfortunately. But you can see in this mid-row, Birch Head, we got temperature data for all 3 years. So I'm going to zoom in on just that location. And these 3 plots are all exactly the same. I just have the different years highlighted for each panel. So the gray lines are the daily mean temperatures retrieved from the light trap logger. And the green line is just whatever year is highlighted for that panel. The dashed line are the min and max for all 3 years combined. And this red horizontal line is just showing where 16 degrees is. And that is because several studies, such as the one on the upper right, have found that eggs that are incubated at 16 degrees or above experience greater embryo mortality, they have reduced hatching success, and they have accelerated development. And Singh and his team produced a publication in 2023 that found that Cherry Point embryos did not exhibit a higher tolerance to heat stress compared to earlier spawning stocks, and they used the Port Gamble stock for that. So what this means is that the Cherry Point stock, because they have this later time that they deposit their eggs, they're already experiencing higher temperatures than other stocks of that lay their eggs earlier in the season. So by late spring to early summer, when these eggs are on the shoreline, these temperatures are already warmer. So they might already be closer to their thermal tolerance level, or to that threshold. So if we look at these results, we can see there's a lot of annual variation. And in 2021 in particular, that really stands out to me because it's a lot lower than the other 2 years. And in 2023, you can see that that green line gets pretty close or crosses that 16-degree threshold. Yes. So in summary, Birch Bay is obviously an important habitat and nursery for many different species of larval fish and invertebrates. The phenological diversity, which just means having the presence of these fish that spawn at different times throughout the year, especially for herring, is really important and provides these multiple resource waves for the different predators that rely on them. And the disappearance of this later spawning Cherry Point stock is or will have effects throughout the entire marine food web. And we've seen this in the decline of the Chinook salmon. We've seen it in the absence of surf scoters over the last several years at Cherry Point. And future questions, considerations, are warmer temperatures to blame for the lack of recovery for the Cherry Point herring? Are these eggs just deposited on a beach during a time when they're already kind of near their temperature tolerance? Or is it the many potential stressors or combination of stressors that exist in the area? Was it the historical commercial fishing pressure that the Cherry Point stock, for whatever reason, couldn't bounce back from? Is it the presence of vessel traffic in the area? Is it the cumulative effect of oil spills in the area? Is it a change in their prey availability or the timing of their prey availability? Could it be disease, pollution from storm water runoff? I think it's important that we continue the fight to figure out why the Cherry Point stock has declined so much and has not recovered, as there's so many other stocks in Puget Sound that could face a similar threat if we can define what that threat is. And I think it'd be interesting, due to the presence of so many other larval fish species around Birch Bay, it would be interesting to see if any other species are facing a similar decline. And I just want to give a shout out because this project has been a huge team effort. It takes a lot of field work. We're out there every 2 days on the water in rough conditions. We've had rain and sun and whales and good times and bad times. But I wanted to thank the Lummi Natural Resources staff. I wanted to thank Todd Sandell and Aaron Jacko and the rest of the WDFW Forage Fish Unit, and former DNR staff Betty Bookheim and Aaron Skare, and the DNR Orca Straits District staff who came out and helped us on the boat several times, and for the many past seasons of the Puget Sound Corps who've been essential to this project. So thank you.
Thanks so much, Allison. That was really great to hear. I mean, tough to hear that we have such a decline, but really excellent presentation. Thanks for breaking it down so well. I'm going to start reading through questions in the chat. Our first one, Rick is asking, if we notice a spawning event in the reserve, where should we report it?
That's a good question. I would say WDFW is the best best people, especially Aaron Jacko. They are the ones that are doing those spawn surveys. And if they get reports of a spawning event from like a visual source, they will go up there and sample that in that area. And like I said, those eggs are deposited for, roughly 10 days. So as long as you get that news to them, they'll have time to get up there and make sure they document that presence. We'd love to know about it too, but they're probably the most important people.
Okay. DFW, got it. All right, next up, Kim. I wonder where all of those sand lance are spawning. I don't think many eggs had been found along the Cherry Point Reach.
Yes, that was one of the really interesting things we found in that first year. If you look at our forage fish results, we have a map online. I think we have found 2 sand lance eggs, from our multiple years and site visits along the entire Cherry Point Aquatic Reserve. So finding 2 eggs of sand lance versus the amount of larvae and juvenile that we found in these light traps, I think means that we're missing these eggs when we sample forage fish. I think we're not hitting the right tidal height for forage fish in this area.
Got it. If anyone wants to raise their hand, we can also do some questions from that. Allison, I did have a question. Do you know of any work that's being done up in Canada for those other late spawning stocks? Yeah.
That's a good question. And I did come prepared for that. You can still see my slides, correct?
Yeah.
So I pulled these data from DFO, which is the folks who do herring monitoring up in Canadian waters. And they have 5 groupings of herring that they manage. And the figure on the right here is just showing their spawn index for each of these regions. So the Strait of Georgia are going to be the closest region to Cherry Point. And these figures on the left are showing their spawning dates. So the bottom is showing all spawning dates for all of these 5 stocks. So these are all herring within British Columbia waters. And so you can see mostly they are these earlier spawners of February to March. There are this little cluster, especially in the 1970s, of some late spawners. But when you pull out just the Strait of Georgia stock, which is, or region, which is on the upper plot, you can see there really, throughout time, there hasn't been in big presence of any late spawning herring through the data they collect. I can't speak to their methods, but I can speak just to these graphs I've pulled off their website.
Got it. Thank you. Yeah, a comment from Eleanor. Comment on temperature again. Our state water quality standards consider 16 degrees Celsius to be excellent quality, but looks like there's evidence from Brooke Love et al. From a bit Western that this is not the case for herring. do you want to speak at all about kind of like temperature tolerance of herring, if you have a sense of that?
Well, like I said, let's see, let me go back to this. So there has been several laboratory studies that have looked at incubation at different times, or different temperatures, and for different lengths of time. And so obviously spending more time in these higher temperatures is going to be more detrimental. But it's common that 16 degrees has been this sort of initial instance. Well, anything above 10 degrees, actually, in a lot of these studies have noted that there are these negative effects to the eggs that are on the shoreline. So these results are attached to our light trap, right, which is offshore in about 12 feet of water. And so I suspect that the temperatures that these eggs are experiencing in the very shallow nearshore environment are actually even warmer than what we're showing here. But these are daily means, so it would be interesting to parse out, you know, sort of the length of time that these temperatures are above 16 degrees. You know, are they only hitting it and going back down, or are these temperatures persisting for a longer period of time? And I think it would also be really interesting to get a temp logger out in that near shore vegetation area of Birch Head and keep it out there. And maybe that's something that we can do at DNR.
Cool. Is DNR planning to continue this larval light trap?
Oh, there's the question. We have very limited funding this year, unfortunately, so it's yet to be determined. But there's a strong interest from the Lummi Natural Resources Department and WDFW to keep this study going in some capacity. So that's yet to be determined.
Got it. Okay. Again, if anyone has a question, feel free to raise your hand or put it in the chat. Give it a little while longer. All right, looks like that might be it. Thank you again, Allison. That was a great presentation. Oh, wait, sorry, one more message. How long ago did the DNA for Cherry Point herring happen?
Do you mean like when we got it analyzed?
Yeah, Diane, if you want to unmute and clarify, or clarify in the chat. When it was first analyzed.
We collected those samples in the 2022 season, and then we sent them in for genetics, you know, 6 months later ish. That makes sense. That helps.
Yeah. Alrighty, if there are no other questions on Cherry Point herring. Oh, so Diane is asking, so how do you know they're Cherry Point?
We know that they're Cherry Point because the Cherry Point stock are genetically distinct. And so we sent them into a genetics lab to get the genetics analyzed. And that's when that Eleni Petru, who did all the genetic stock structure for herring in Puget Sound and beyond, she analyzed our results for us, because I'm certainly not a geneticist. So she was able to confirm that through the analysis she ran, that 3% of our samples were from the Cherry Point stock.
Okay, got it. Dan, hopefully that answers your question. Sweet. All right. Well, I am going to share my screen briefly just to wrap us up. Thank you again, Allison. Sorry, share screen. More. Oh yeah, Allison, sorry, before I wrap up, this question from Lyle, maybe you could touch on. Are the Cherry Point herring not on the ESA because of the overall herring populations?
Yes, that is true. There were petitions to get it listed multiple times, and the folks in charge decided that although Cherry Point stock was different from others, it wasn't a detriment to the species as a whole. So they couldn't classify that as a, forget what they call it, like a separate species segment or something like that.
Okay.
Unfortunately.
Yeah. All right. Thank you. Okay, so thank you all for coming. I'm just gonna wrap it up here. Thank you so much to all of our presenters, and as well as the Citizen Stewardship Committee, and thank you all for coming. I especially want to thank Robert and Lyle for helping me get this event organized, and also just thank you to Olivia and Lindsey and Leah and Allie and Allison for such informative presentations. If you, after going away from this, are feeling like you want to protect the Salish Sea, one thing that would be great, part of my AmeriCorps, is getting pledges of community members that are pledging to protect the Salish Sea. So I will have Eleanor put that link in the chat. It's a really quick survey, just how you're going to help protect the Salish Sea, and that helps me and resources. If you are interested in getting involved with the Citizen Stewardship Committee, feel free to reach out to us at this email, aquaticreserves@resources.org. Specifically, we have this upcoming event, our Cherry Point Aquatic Reserve CSC meeting / training will be on May 1st will be a Zoom session, and then May 8th will be an in-person. And that will be a European green crab molt search training, and that's using the MyCoast app. More info about resources can be found on our website. Resources depends on donations to keep putting on events like these. So if you have a little bit to give, please consider doing that after this talk. And lastly, you will receive a follow-up email with the recording of this event, as well as a lot of the links that were shared in the chat by both us at Resources, as well as folks like Lindsey with that cheat sheet, and just other links that came up as we were going through. Thank you all again so much for joining us on Zoom. One of the nice things about this event now being on Zoom is that we can stay inside on these very, very cold days. And so I hope that you all have a wonderful rest of your Saturday and that you stay warm. All right. Thank you all for coming.